Hello everyone, welcome once again, and always my pleasure to talk about the important portions from the biology chapters. Today the talk is about the application of biotechnology in agriculture. So the huge development, say from green revolution, there has been a significant and very important developments in agriculture. But mainly it is because of the use of fertilizers and chemicals. But we also have to think that these fertilizers, chemicals are causing toxic effects in the organisms who are consuming these crops and plants. This is called in biology the biological magnification, which is the maximum in humans because we are consuming these crops the most, right? So how to reduce the use of these chemicals and fertilizers, that is a $1 million question. Now the role of biotechnology comes here, and it plays a very important role because of the benefits that it provides in the agriculture segment. Actually, what is being done in biotechnology is by manipulation, the genetic constitution is modified in the organisms, and they are called the genetically modified organisms, GMO. So in the plants and crops also this is being done, and the benefits that we get is the plants become pest resistant, most importantly, because the huge losses are being incurred because of the pests and insects, and pesticides and insecticides are being used. So it is a very big benefit if we get pest resistant crops, and the crops become resistant to specific pests without the use of insecticides and pesticides. Then number two benefit is that the use of minerals become more efficiently, it is done. The minerals are used by the crops or plants more efficiently, so the fertility of the soil is retained. So this is also a good big benefit, and special quality of crops or yield can be obtained, like the golden rice, which is enriched in vitamin A. This is also a contribution of biological biotechnology, right? And then the crops become more resistant to the adverse effect of the abiotic components like the drought, maximum heat, excessive heat, salt, etc. So these are the benefits, but the most important benefit of these biotechnology applications is the production of pest resistant crops. So actually what is being done is basically you have to know that there is a bacillus, a bacteria which is known as your in short it is BT. The full form is the Bacillus thuringiensis. Bacillus thuringiensis is the full form of this bacillus, and in short it is known as BT. So it produces a toxin which kills the insects and pests. These toxins are nothing else, are protein crystals. But question is, if the toxin is killing the bacteria or the insects, killing the insects and pests, then why it is not killing the bacteria itself? So the answer to this question is that in the bacteria, when it produces this protein crystals in the toxins, it is in the inactive form. Okay, it is protoxin. But when it gets inside the insects or pests, in the gut, it gets the alkaline medium, and this toxin becomes active and kills the insect or the pest. So this is why it does not kill the bacteria Bt itself, but it kills the insects and the pests. So this is very clear now, I think. So this is done, and this technology is also called Bt technology. So the toxins of the bacillus, of this bacillus Bt, is cloned and is expressed in the plants and crops, which become pest resistant or insect resistant without the use of insecticide and pesticide. Right. So these toxins are insecticidal proteins, in fact, protein crystals. So they become active in the alkaline medium in the gut of the insects, and they kill the insects because they are insecticidal. Okay. Now the specific encoding of these genes of the toxins of Bt are applied in different crops which are destroyed by the pests like cotton, like tobacco, etc. And the different germs, for the specific germs, there are specific encodings, okay, in the genes, like Cry1AC, Cry2AB. Okay, so Cry1AC is for the cotton bollworms, which affects the cotton mostly, and Cry2AB is for controlling the pests, corn borer. So these are the encodings of the genes, right? And pest-resistant plants are also being formed by this biotechnology devices, like RNA interferences are being done in the case of these worm-infested plants. In tobacco, maximum takes place mostly. Maximum cases it is found that tobacco roots are being destroyed by this nematode, okay, these worm. And here what is done, the RNA interference process of RNA interference, the RNAi, which is used for the cellular defense, it is converted normally into a mRNA, which is silenced, which is not allowed to convert into mRNA, and then the defense process is lost. Okay, the RNAi is not effective, and it is not being converted into mRNA also. So this destroys the cells of these very, very harmful worms, which destroys the crops. Mostly we are taking the example of tobacco. So using this agrobacterium vectors, nematode specific genes were introduced into the host plant. The introduction of DNA was such that it produced both sense and antisense RNA in the host cells. These two RNAs, being complementary to each other, formed a double-stranded dsRNA. This is the complementary double-stranded RNA which is silencing the mRNA. So this is the process which is being done in the post-resistant plants. So in detail you will study further. So I think I end my topic here only. So in the next videos I will come with other important topics like your gene therapy, etc. Thank you so much. God bless you all. If you like my videos, please subscribe to my channel, and also write to me if you want to know anything else regarding these topics. Any question, any query, please let me know. Thank you so much. God bless you all.